What's going on, everyone? Today we are going to be finishing up our lecture on metabolism, and so we will be discussing how we actually get energy in our cells from the food that we consume. And so we are going to go through the processes of how we get ATP, both through glycolysis and aerobic metabolism as well. A big reason why we consume oxygen is because it's part of our metabolic process. So cellular respiration, that is going to be the breakdown of nutrients to generate ATP in the presence of oxygen. So we consume foods, they have carbohydrates, they have fats, they have proteins, and then within the cell, we're going to use oxygen in order to generate energy. And so once we metabolize those nutrients, our byproducts are going to be carbon dioxide, which we exhale out, water, which is, of course, makes up a lot of our body composition, but we also exhale and get rid of it in other ways. And then energy, which is what we're looking for. And so this ATP that's generated from these glucoses, fatty acids, amino acids, that's going to be the energy used to carry out other chemical reactions within our cell. Our cells are going to break down glucose, fatty acids, and amino acids in order to get ATP. So glucose is broken down through a 10-step process called glycolysis, which we'll talk about in a moment. But the byproduct of glycolysis, right? So this is anaerobic metabolism right here, breaking down glucose to pyruvic acid, which we get some energy from. But the byproduct, pyruvic acid, in addition to fatty acids and amino acids, can be broken down through aerobic metabolism. And so if we have oxygen, it's going to drive the breakdown of this acetyl-CoA through Krebs Cycle, and then some of those byproducts through Krebs Cycle and glycolysis will be used to generate energy in the electron transport chain. So that's a general overview, but let's break it down a little bit more. This image below shows the metabolic processes that are occurring within our cell. So we can notice that this is a mitochondria outlined here, and that glucose is broken down in the cytosol outside of the mitochondria. So we get some energy through glycolysis, that breakdown of glucose. But then also what happens along the way is that we have what's called electron carriers, which are going to help carry out this conversion from glucose through this 10-step process to pyruvic acid. And these electron carriers, they help out in glycolysis, Krebs cycle, the formation of acetyl-CoA, and all of those electrons are going to be transported to what's called the electron transport chain. And this is where we are going to generate a majority of our ATP. And that's why we call the mitochondria the powerhouse of the cell, because we're getting most of our energy through the aerobic metabolism that's taking place here. And so the byproduct of glycolysis, pyruvic acid, and like we said earlier, fatty acids and amino acids can be converted to acetyl-CoA, go through Krebs cycle process, and then eventually generate ATP to carry out certain functions within our cells. In order to show all the metabolic pathways, what I'm going to do is start with one glucose molecule and take you through all of the steps that can be used to convert that glucose molecule into energy. So that carbohydrate metabolism, what we're doing when we break down carbohydrates is we're trying to get glucose, and that glucose will be used within our cells in order to generate ATP. So first we have to go through glycolysis, and we're going to break that down. Then we need to convert pyruvic acid to acetyl-CoA, and then acetyl-CoA can be used to generate more energy through the Krebs cycle and the electron transport chain. Glycolysis is anaerobic metabolism, meaning that you do not need oxygen in order to break down glucose to pyruvic acid, but then pyruvic acid can be used in aerobic metabolism when we convert it to acetyl-CoA. But first, let's go through just the breakdown of one molecule of glucose into pyruvic acid. And so here you can see that there are 10 different steps to convert glucose to pyruvic acid. And so these are 10 different enzymes that are going to make this change from glucose to all these intermediate products to pyruvic acid. During glycolysis, what we are going to do is break apart chemical bonds and add and subtract phosphate groups in order to generate energy and then to get pyruvic acid, which then can be used later on through aerobic metabolism to generate even more energy. But this whole process starts with one glucose molecule, and it's going to go through the first enzyme, which is hexokinase. And hexokinase is actually going to require we use one ATP molecule in order to convert glucose into glucose 6-phosphate. So if you're keeping record, we're already down one ATP after the first step of this process. But don't worry, we'll make up the energy later on. Next, we are going to convert this glucose 6-phosphate into fructose 6-phosphate, and phosphohexose isomerase will be the enzyme that does that. Once we get this fructose 6-phosphate, we are going to add another phosphate group onto this molecule, and the enzyme that does that is phosphofructokinase. So now, again, we're going to use another ATP to add another phosphate group onto this molecule. So after step 3, we're down 2 ATP. But again, we'll make it up later on. So now in step 4, the 6-carbon fructose 1,6-biphosphate has to be converted into 2 3-carbon molecules called glyceraldehyde 3-phosphate. And before we get to 2 of these, one of these 3-carbon molecules is actually going to be dihydroxyacetone phosphate. And this needs to be converted into glyceraldehyde 3-phosphate through an enzyme called isomerase. And so now here we have 2 3-carbon molecules, each with a phosphate group on them, and they're going to continue along in glycolysis. Enzyme 6 is going to be glyceraldehyde 3-phosphate dehydrogenase. And the dehydrogenase comes from the fact that an electron carrier and a cofactor for that enzyme named NAD, or nicotinic adenine dinucleotide, is going to come in and actually oxidize this molecule. And so when it does that, it's going to pick up two hydrogen atoms. And with those atoms come protons and electrons. And so those NADs are going to pick up those hydrogen protons and the electrons, and the NAD now converted into NADH is going to carry those electrons to the electron transport chain. And again, we're going to break it down a little bit further, but you're going to see through these metabolic processes that 2 electron carriers, both NAD plus and FAD, are going to be the ones that generate ATP. So as we go along through these metabolic processes, keep in mind that for every NAD and FAD that picks up hydrogens and electrons is going to be converted into energy. Each NADH plus H is going to generate 2.5 ATP in the electron transport chain, while each FADH2 is going to produce 1.5 ATP. We're going to be discussing this a little bit later on, but the reason is because they're going to be donating these electrons and hydrogens to the electron transport chain, which will then give us enough energy to synthesize an ATP. But all along these reactions, we're going to see that a lot of this ATP yield is going to come from the fact that these electron carriers are going to transport all these electrons and all these hydrogen protons to the electron transport chain. So now going back to glycolysis, we're going to see that during this step, not only did we give those electron carriers some electrons, but we're also adding a phosphate to each one of these 3-carbon molecules. So now we're left with 2 molecules of 1,3-biphosphoglyceric acid. And then an enzyme, phosphoglyceric acid kinase, is going to actually take one of those phosphates and add it to an ADP. And now since we have 2 molecules, we've gained 2 ATP. So now finally, we lost 2 ATPs earlier, one in step 1 and one in step 3, but now we've gained 2 of them back. So for a net so far, we've gone through 7 enzymes and we haven't made any energy. So what we're going to see for steps 8 and 9 is that these enzymes are going to basically change the conformation of these 3-carbon molecules and change the location of the phosphate group. And then here, step 10, enzyme 10, pyruvate kinase, what we're going to see is we're going to cleave off those phosphates, and those phosphates can be added to ADPs to now make ATPs. So in this last step, we got 2 more ATPs. So that whole process, the conversion of 1 glucose molecule to 2 molecules of pyruvic acid, we netted 2 ATP, which doesn't sound like a lot, but also remember, we have these electron carriers, these NADH plus H's, which are going to carry electrons through the electron transport chain, which will then be used to synthesize more ATP. In addition to that, we could use these molecules of pyruvic acid to also get more energy through aerobic metabolism. Here we see that pyruvic acid is going to have 2 different routes. Either we have oxygen, an ample amount of oxygen to go through the aerobic pathway, or if we don't have enough oxygen, that pyruvic acid can be converted into lactic acid. And so when you're going to see lactic acid buildup is when people are working out at high intensities because they're not able to get enough oxygen to convert all these pyruvic acids through the aerobic pathway. And so some of those pyruvic acids get converted to lactic acids, and that can actually start entering the bloodstream. But, for the metabolic pathway here, these 2 pyruvic acids that we got through glycolysis, as long as there is oxygen, then they can be shuttled into the mitochondria, and this pyruvic acid is eventually going to be converted into acetyl-CoA. But now I want you to remember, because we're trying to keep track of all the energy we're going to get from one glucose molecule, that during this process of pyruvic acid to acetyl-CoA, you see this NAD is going to come pick up electrons, and this is going to be used again for energy later on down the road. It's important to note that acetyl-CoA is not only going to come from pyruvic acid, it could also be derived from amino acids, fatty acids, which are going to go through beta oxidation in order to form acetyl-CoA, and also ketone bodies. This acetyl-CoA is what's going to be used in aerobic metabolism. So all of these molecules, all these nutrients need to be converted into acetyl-CoA before entering the Krebs cycle and getting more energy out of these molecules. Here we can see that acetyl-CoA is going to enter what's called the Krebs cycle, or otherwise known as the citric acid cycle. And so remember, from our original glucose molecule, now we're left with 2 acetyl-CoAs. So everything we get through this process, you're going to multiply by 2. And here we're going to see that on step 3, step 4, and step 8, we're going to have NADs come, pick up electrons, and then dump them off at the electron transport chain. So again, trying to keep track of all the totals, through the Krebs cycle, we are going to get 6 NADHs plus H's. We also see on step 6 right here that we're going to have FAD come in and pick up electrons and hydrogens to be used for energy at the electron transport chain. So we have 2 of those. And then also we get 1 ATP molecule per acetyl-CoA on step 5. So the grand total, 6 NADHs plus H's, 2 FADH 2s, and then 2 ATPs are what we're going to get from our original glucose, our original glucose molecule being converted to acetyl-CoA and then going through the Krebs cycle. So we talked about NADs and FADs and them picking up electrons and picking up hydrogen. So where are they going? They're going to what's called the electron transport chain, which is found on the inner membrane of the mitochondrial matrix. And so where are we actually getting the energy to generate ATP from? Remember that we have 2 types of energy. We have kinetic energy and we have stored energy. So we're going to use the movement of protons and the concentration gradient between the mitochondrial matrix and the intermembrane space in order to generate ATP, to get the energy necessary to wind up with ATP. What's going to happen is that NADH plus H is going to come and drop off its protons and its electrons at the electron transport chain. And as these electrons move from complex 1 to complex 3 to complex 4, there's going to be a certain amount of hydrogen protons pumped from this area of low concentration to this area of high concentration. And those electrons are needed in order to do that because it's very hard to get molecules to go from an area of low to high concentration. They want to move in the opposite direction, an area of high concentration to low concentration. So those electrons provide that energy needed when they move in order to have that proton pumped. And so we get 4 protons that are pumped in complex 1, 4 protons that are pumped in complex 3, and then 2 protons that are pumped in complex 4. And so we're going to have 10 hydrogens now on this higher area of concentration, this intermembrane space. But now what's going to happen is that since we made that gradient a little bit bigger, now we have the ability to pump these hydrogens back to the space and to the matrix and use that energy in order to synthesize ATP. And so these hydrogen protons, they are going to be attracted to that oxygen, and that's why we needed oxygen for all of this to occur. Oxygen is going to be used to accept electrons from this electron transport chain and hydrogen protons in order to form water. And so what's going to take place is that basically we are going to have these hydrogens, shoot through this 5th complex called ATP synthase. Those hydrogens will bind to oxygens, and then that energy that was gained from the movement of the proton, that's going to be the energy needed in order to bind a phosphate to ADP to get ATP. And so with NAD, since it drops its electrons off at complex 1, and overall it's going to pump 10 protons. Well, we're going to need 4 protons pumped per ATP. So that's why, if you're wondering where do we come up with this 2.5 ATP number, that's because, we pump 10 protons, so that makes 2.5 ATP. It's not going to come up as half an ATP. It's just the energy needed in order to form 2.5 ATPs. The difference is for FADH2s that it's not going to drop its electrons off at complex 1. It drops its electrons off at complex 2. And then as those electrons go to complex 3, 4 protons are pumped here and 2 are pumped here, giving you 6 protons in total, and that's going to give you 1.5 ATP. So this process, right, as long as we have oxygen, all of those substrates from earlier, the pyruvic acid, the beta oxidation from the fatty acids, can then go through Krebs cycle, and then all of those electron carriers can then dump off their electrons and those hydrogens in order to form ATP. And this is where we're going to get a majority of that ATP from our original glucose molecule. If we use those 2.5 and 1.5 numbers from NAD and FADHs, we could see that our total yield from one glucose molecule is going to be 2 ATPs from glycolysis. Those are anaerobic metabolism. We just went ahead and got 2 ATPs for then the cell to use. But then these NADHs that came during glycolysis, they're going to generate roughly 5 ATP. The NADHs that were produced through the conversion of pyruvic acid to acetyl-CoA, those will also produce roughly 5 ATP. And then the Krebs cycle, we got 2 ATP, 6 NADHs, which are going to provide roughly 15 ATP, and then 2 FADH2s, which provide roughly 3 ATP. So if we take this, will be 18, 20, 25, 30, 32. So that is our yield from one glucose molecule. If you took one molecule of glucose, took it through all the steps, as long as there was oxygen present, you would yield roughly 32 ATP molecules. So that was all starting with one glucose molecule. If we started with lipids or our triglycerides, if we broke those down from our fatty acid cells, we could break that down into glycerol, which can be used as part of glycolysis to generate ATP. But those fatty acids will go through a process called beta oxidation, and that's going to be the conversion of fatty acid to acetyl-CoA. And then we're going to yield all the same things that we did from that glucose molecule, right? Because again, this byproduct, this intermediate here, is going to be exactly the same. So we'll yield from that point on, from the Krebs cycle and electron transport chain, we'll yield the same amount of ATP. We can also break down amino acids in order to get energy. And so those amino acids are going to be hydrolyzed. to get ammonia and a keto acid. The ammonia is going to go through the urinary system, and we're going to expose of it. But that keto acid can be either directly converted into acetyl-CoA or converted into pyruvic acid, which can also be converted into acetyl-CoA, or turned into a Krebs cycle intermediate called alpha-ketoglutarate. And if we go back to our Krebs cycle diagram here, we could see right here, right before step 4, that could enter the Krebs cycle, and then we'll be able to yield some ATP from that. Here you can see the different amino acids and where in the Krebs cycle that they can be converted to. So a majority of them can be converted directly to acetyl-CoA, but other amino acids can be converted either to make alpha-ketoglutarate or succinyl-CoA or fumaric acid or oxaloacetate. And so these are different ways that amino acids can enter the Krebs cycle, and we could yield ATP, we could yield energy from them. So now that was the cellular breakdown of everything, and that's going to be very important for you guys to go over and memorize and read in your textbook. But I do want to just sort of, zoom out for a second and just talk about overall metabolism, and we're going to talk a little bit more about that in the next lecture as well. But we have nutrients that we consume day to day, and we have a recommended amount of nutrients given to us by governing bodies. But nutrients are going to be chemical substances in food that the body uses for growth, maintenance, and repair. And so we've talked about many of these already, but the 6 main types are water, carbohydrates, lipids, proteins, minerals, and vitamins. Water obviously is a very important element since most of our cells are made up of water, and much of our functions within our cells and in our bodies involve water. Carbohydrates, lipids, and proteins are all energy sources, but as we know, those cellular molecules of glucose, fatty acids, and amino acids can be used for other structural things in the cell as well. And then we have vitamins and minerals, which we talked about earlier in the last video, that can be used by enzymes in order to carry out reactions. And so minerals are going to be inorganic elements, things like sodium, potassium. Many of them are going to carry a charge. Magnesium. There's many different types of minerals that we can consume or found within our bodies. A lot of them are concentrated within our skeleton, within our bones. But they also, those ions are used to regulate enzymatic reactions, and many enzymes are going to need these cofactors in order to carry out reactions. So again, we do have vitamins, which are organic. So they have that carbon backbone, and again, we're going to classify them. They are cofactors, but we classify those organic cofactors as coenzymes. And that's why it's important that we recommend a certain amount of vitamins and minerals every day. So we have fully functioning enzymes, and they're able to carry out reactions in our cells. That is going to conclude our metabolism lecture for today. We are going to talk a little bit more about gross metabolism in the next video, but today we went over glycolysis and electron transport chain and, Krebs cycle, citric acid cycle. So these are very complex pathways, and just reviewing the video one time probably won't be enough for you to get the overall grasp of it. So make sure you're reviewing the video, reading up on textbooks, checking other resources to make sure you're understanding what's going on in the cell and how we actually generate ATP. Thank you guys so much, and I'll see you next video.